Okay, so first I would like to thank very much Professor Sohail for inviting me here and the possibility to talk a little bit about very snakes and snake bites in Roraima. It's a very difficult name to say in English, but Roraima is this one of the most northern states in Brazil. And this is the place where we have the highest incidence of snake bite and lethality. And also we have a lot of different ethnic indigenous populations. And in this group, the incidence of snake bites is even 12 times higher than in the normal population. So it's a place where snake bites are really important. And also different, we don't have much rattlesnakes. We don't have almost rattlesnakes in the Amazon. But here in Roraima, we have a subspecies, Crotalus durissus ruruima, which is responsible for 11% of the human accidents. And as I said, there's not much rattlesnakes here. And most of the rattlesnakes in the south of Brazil, they have neurotoxic venoms, type 2 venoms. And in Roraima, there's a lot of clinical reports showing that they might have type 1 cytotoxic or hemorrhagic venoms. And doctors stand there to press Butantan to send Botrops crotalus antivenom, which is a mixture. And we don't have much evidence to support the use of this mixture of antivenoms there. And then saying that antivenoms in Brazil for rattlesnakes are made only by type 2 venoms. And then we aimed, working with the Roraima people, we aimed to check, okay, do we really have type 1 venom? The only proteome we have from this species is from 1, and it's a type 2 venom. And then, is there type 1 venom in Roraima? And do we really need to use Botrops, the mixture Botrops crotalus venom there for the human accidents? And for that, we did a collection of 19 samples of snakes, most from the northeast of the state, 14 adults, 5 juveniles, 12 yellow and 7 white venoms. They play with this different color of rattlesnake venoms. And with these venoms, we did proteomics and functional activities, and also we tried to neutralize the most important toxic activities with Crotalus antivenoms, which is the rattlesnake, Botrops antivenoms, which is the jararaca Botrops venoms, and a mixture of those. So the first thing we did was individually to compare by the classical reverse phase HPLC C18 columns. And here we found 4 venoms that behaved exactly as we expected for type 1 proteolytic or hemorrhagic venoms with the highest peak containing SVMPs, and 11 venoms containing the highest peak here with crotoxin. It's not all type 1 or type 2. We found also 4 venoms with a mixture of both. So we did a pool of type 1 and type 2 venoms and went for the toxic activities. So the first one was the hemorrhagic activity, the most that the local doctors tried to push us to send the Botrops antivenom. And indeed, the type 1 pool presented highest hemorrhagic activity, although much less than Botrops jararaca venom. On the other hand, comparing myotoxic activity, type 2 was more myotoxic, similarly to Crotalus durissus terrificus, and type 1 much less, just like jararaca. procoagulant activity was quite similar, and the lethality was, as expected, much, much higher on type 2 venoms than on type 1. And then we went to the proteomics of individual samples. And what we see is that 7 isoforms showed statistically significant difference between the type 1 and type 2 pools. And these 4 isoforms, this is very low, but this is one PLA2 unit of crotoxin. And these 3 were 2 SVMPs that were predominantly expressed in type 1 venoms and 1 PLA2 in type 2 venoms. We did then, we tried to correlate the presence of these isoforms with several situations. I'm showing here 2 where we got some indicative results, like the ontogenetic stage of the snakes. Here in pink is the adult snakes, and in green, the juveniles. And we can see that the PLA2s are predominant in juveniles, while SVMPs were similar, predominant in adults. But here, we see that both juveniles and adults show both types. It's not exclusive of that type of snake, but there is some tendency to type 2 venoms predominate in juveniles, showing ontogenetic evaluation of this venom. With geographic distribution, we found something interesting. In central area of the state, we got almost only type 2 venoms with just a single cluster here with high phospholipase. While in the Northeast, we have bimodal distribution showing both type 1 and type 2. Here, in the central area, high phospholipase, low SVMP. The sequence, this is one of the phospholipase that matched almost perfectly with the acidic subunit of crotoxin of Crotalus durissus terrificus venom, with just one substitution, exactly in the cleavage site of the gamma chain. I don't know the functional meaning for that, but in the Western blot, we see that it might be happening. And in the B subunit was exactly the one, subunit CBC, isoform C from Tsabikan, one substitution, subunit D, and a little more with subunit B and A. So in this case, we do expect that heterologous venom made with Crotalus durissus terrificus will bind and neutralize due to a lot of similarity between the sequence in Crotalus durissus terrificus. In the case of the SVMPs, it was the same. The most dominant in ruruima, presented just one substitution with the SVMP from Crotalus colilineatus. However, in the pool of venom used for immunization to make antivenoms, we don't have many SVMPs. And then this antivenom doesn't recognize many SVMPs. And then we compared with the SVMP predominant in the pool of venom used to prepare Botrops antivenom. And we see here in the bottom line, a lot in red, a lot of amino acid substitutions, especially in the region coding for the catalytic domain. So we ask, in this case, would Botrops neutralize, Botrops antivenom would be effective in neutralizing type 1 Crotalus SVMPs? And then we went for the reactivity with antivenoms. And here we see the reactivity by ELISA, Botrops, Crotalus, and the mixture with type 1, type 2, and the controls. And Crotalus antivenom did react better with both types of ruruima venoms. And by Western blots, Crotalus antivenom did not recognize SVMPs by type 1 or any other venom. And Botrops antivenom recognized very well the SVMPs here, P3 and P1, but did not crotoxins. And the only that recognized all of them was the mixture. However, when we went to the neutralization of toxic activities of the venoms, here trying to neutralize the type 2 neurotoxic venoms. In blue, we have the neutralization by Crotalus antivenom, in red by Botrops antivenom, and in yellow, the mixture Botrops and Crotalus antivenom. We see here lethality and procoagulant activity and myotoxic activity very well neutralized by both Botrops and the mixture, while the Botrops antivenom did not neutralize any activity of type 2 neurotoxic venom. When we go to the type 1 hemorrhagic venom, we find something a bit similar because lethal activity was very well neutralized by Crotalus antivenom and the mixture, while Botrops antivenom neutralized only partially the lethal activity even of type 1 venom. Procoagulant activity was even more interesting because Crotalus antivenom and the mixture completely neutralized procoagulant activity of type 1, and Botrops antivenom did not have any effect, showing that SVMPs working in the procoagulant activity were completely different between type 1 Crotalus and Botrops snakes. The only thing that the Botrops antivenom was a little bit better than the Crotalus antivenom was in the neutralization of hemorrhagic activity. But I want to remind you that it was a very mild activity even in type 1 venoms. So as conclusion, we do have type 1 and type 2 venom phenotypes in Roraima. Juvenile snakes apparently display type 2 phenotype predominantly, and then this ontogenetic variation that happens in other Crotalus species may be happening here as well, but we need more. The number of snakes was still very low to assure that. And also type 1 phenotype was primarily found in the region bordering Guyana and Venezuela, where we have other Crotalus species that do have type 1 venoms. And the presence of type 1 and type 2 venoms in Roraima may explain the difference previously observed in human envenomings. Concerning the antivenoms, but we do not recommend the use of only Botrops antivenom, which some doctors have this in mind there. So please don't do that. We do believe Crotalus antivenom will be sufficient to neutralize all the accidents. However, we do need some trials and some observations to make sure about that. On the other hand, Botrops Crotalus antivenom works against all the types. So it should be used. They should continue to use that. However, we have to have in mind, I would like to see these studies with Crotalus antivenoms done. Why? Because when we give the mixture, we give a lot of heterologous antivenom that we know are not doing anything against that venom. So we can do that, it will neutralize everything, but we might be doing useless proteins to human accidents. So to finish that, I want to acknowledge the people that helped in this work, and the people from Butantan, people from University of Roraima, and the team of CONFAP, which is this program that I acknowledge very much. It is a joint program of lots of Brazilian supporting agents for science, and they made this initiative just to enhance the research in the Amazon. And thank you very much for your attention.